702. Let's see for 5 more minutes if anybody else joins, otherwise we'll start. Okay, let's start. So, okay, I'm Tanishka, and let's see what we have to go through today. So, in all our previous classes, we have studying the basics of immunology. So, immunology is the study of the immune response which our body performs against a foreign pathogen or antigen. Okay, so and there are certain type of cells which take part in this immune response process, and those are immune cells. So we have seen that there is a innate immunity and adaptive immunity, and suppose one pathogen infects our body, and then the first cell which encounter them are neutrophils. Along with that, there further comes the monocytes and all those cells, and after that there is a adaptive immunity which comes in the picture. So there are specific cells, so that include the T cells, the T lymphocytes and B lymphocytes. Okay, and the antigen which comes here, so the antigen is what happens. So this antigen binds to a set of cells which is called as antigen presenting cells. So suppose a foreign pathogen has come inside our body, and the first line of defense is not able to take care of that particular pathogen. Then what happens, there are certain cells which are taking that antigen, and then they process or present them on their surface. Okay, so that's why, as they are presenting the antigen, they are being called as antigen presenting cells. Okay, and there are professional antigen presenting cells which include macrophages, dendritic cells, and the B cells or B lymphocytes. So these three are antigen presenting cells. Okay, and these antigen presenting cells present antigen to the B or T lymphocytes. Okay, so there are these T cells, sorry, the T lymphocytes, so which are like this T cells and B cells. Okay, so these T cells, they are having one molecule which is called MHC on the, so sorry, so they are having receptors for antigens. Okay, so that these T cells, they are having receptors. So for example, here you can see this is one particular antigen presenting cell. So this is a foreign antigen which is there, and then this particular cell has engulfed that antigen inside their lysosome, and then degraded that and makes peptide out of that, and that peptide is going and being presented on the surface of this particular cell with the help of a surface molecule which is being called as MHC. Okay, so these are MHC molecules which present the antigen, and these T cells, they can interact with the antigen only if they are being presented via MHC molecules. Okay, so we have read that there are two types of MHC molecule, MHC class 1 and MHC class 2. Okay, they both present antigen to T cells. However, the way they process the antigen for the presentation is different. Okay, so for example, this MSC class 1, so they process the antigen, the endogenous antigen, process them and present them on their surface MHC, and that is MSC class 1. However, the MHC class 2, they take the exogenous antigens and then process them and present them with the help of MHC class 2. Okay, so there are two types of MHC, and MHC class 1, they present antigen to T cells which are cytotoxic T cells, and MHC class 2, they present antigen to the helper T cells. These are two types of T cells. Okay. So here this is an antigen presenting cells being shown. Okay, this is a macrophage. This is a foreign antigen which is being inculcated, processed and presented on MHC, and this MHC presented antigen, it is binding with the T cells. So this is with the help of T cell receptor. So the antigen is binding with the T cell with the help of T cell receptor, and after the binding there is a so this is the first signaling 1, and then after the activation this cell start releasing some chemokines, cytokines. That cytokines further signal more and more T cells for being activated, and also this also gives the signals to the B cells, and that's why there is more and more B cell activation is also happening. So at a time there are more than one signal that these and during this antigen and T cell interaction that takes place, which makes the interaction more specific, and that's how the immune response gets activated. Okay, so for now just think like this is one antigen presenting cells, and on the surface of it is having this MHC molecule. With the help of that it presents the antigen. Okay, and this is two types, MHC class 1 and MHC class 2. There are two types. Okay, so the protein product of MHC class 1 and class 2 genes are highly polymorphic. So polymorphic is there are more than one variety of these MHC molecules. Okay, so and why is that? Because this MHCs, so there are only two types, MHC class 1 and class 2, but only these two MHCs are there to present all variety of antigen to our T cells. Okay, so as it has to present a variety of antigen, there should be a polymorphism. So in order to present a lot of or variety of antigens, these MHC molecules are having polymorphism in them. Okay, that means like one variety of MHC molecule, it can present antigen 1. Okay, but another variety of that MHC molecule, that is not suitable for presenting that antigen 1. It only being presented specifically for one variety of MHC molecule. So even though it is class 1 molecule, but there also it has variety. Okay, so these genes, these you can see that DPB and DPA, so and DRB, DRA, and these are alpha and beta subunit of the MHC molecules. Okay, so here you can see there is a 351 a variety of that MHC class 2, this particular gene, and 19 of alpha or DPA, 435 of DQB, 32 of DQA, and 1211 of DRB. Okay, so this shows there is a high polymorphism. That means there could be SNPs, the single nucleotide polymorphism, or any other kind of variety generation. Okay, and in MHC class 1 also, so there also you can see there is variety of these MHCs subunits are there, or the genes that makes these MHCs are there. So for example, here it is shown that total these some 2668 of B gene of MHC class 1, these many varieties are present. So these, not all of these are from one human being. So there are, these data are from whatever pool of MHC we know so now, so far. Okay, so for example, there is one person, so that is having suppose 500 uh, polymorphic MHC class 1 genes. Okay, so that 500, and other person is having 500 varieties. Okay, so total there are 1000 varieties of MHC class 1 which are known to us yet. Okay, so it is not essential that all 2668 varieties are present in one person, but there are variety which is more than one in one person, and all together we know that for this particular gene these many varieties are known, for this one these many varieties are known, and that is due to the variety of antigens that are being presented by MHC class 1. Okay, so this is what is polymorphism, and it is highly present in MHC class 1 and 2 class 2 genes. Okay, and also expression of these MHC alleles is codominant. Okay, so you must be knowing, like in the genetic terms we know that there is a dominant and recessive gene. Okay, so if for one gene there are two alleles, and if there is a dominance and one allele is carrying the dominant copy, another is carrying the recessive copy, the phenotype will be of the dominant one, or like only one will be expressed. But in this case of MHC allele, they there is a codominance. That means here you can see, this is an example. In this individual there are two alleles, two different alleles of the MHC gene. Okay, and on the surface, whatever is being expressed, that will be shown on the surface. So even though like there are two varieties, there is no dominance, there is a codominance. So both of the alleles will be equally expressing on the surface. Okay, for example, this individual also, there are two varieties, and both of the varieties are being expressed. Okay, and also when there is a uh, mating, then after the fertilization, these is being inherited in their children as well. So for example, when these a male and female are having children, both of these their MHC alleles are being transferred in a combination. So and all of them are being expressed in their progenies. So that is how the varieties are being these varieties are being transferred by generation, and also they are having codominance. Okay, so suppose in my body I have 200 types of MHCs, and that means all of those 200 polymorphic MHCs are being present on my antigen presenting cell surfaces. Okay, so the protein products of these MHC class 1 and class 2 genes are highly polymorphic and also polygenic. Okay, so for expressing this MHC class 1, there is not only one gene, there are more than one genes which are expressing. Okay, so this is polymorphism, more than one variety, and there is a polygenic, there are three copies you can see, and all of them are being expressed. And this is polymorphism as well as polygony showing together. So okay, so here polymorphism, more than one variety of the MHC class gene, and also polygyny, that means more than one gene, and as well as codominance, all of them being expressed on the surface. Okay, so MHC polymorphism, it affects antigen recognition by T cell by influencing both peptide binding and the contact between the T cell receptor and MHC molecule. Okay, so this is the uh, MHC, and this is MHC class 1, this is MHC class 2. So we have seen in the previous classes, so it is one single subunit of alpha, alpha 1, alpha 2, alpha 3, and this is the beta. And here we are having alpha 1, alpha 2, and beta 1, beta 2. Okay, so here you can see this, the cleft which is there, this red cleft where the antigen binds. So with the help of this cleft only, the antigen is being presented to the T cell. Okay, so there you can see there is, this part is red, that is present between alpha 1 and alpha 2. Here you can see, so there also you can see there is a beta sheets in the center, and here you can see there are these alpha folds. Okay, and there these red dots are showing the variability. Okay, so how much changes are there, like at what locus there are changes, as we are saying there polymorphism, but at what place. Okay, so that polymorphism is coming majorly here, the amino acids or the gene sequence which is present here in this cleft, because that cleft is where the antigen is binding, and the polymorphism is for that purpose only, so that it can bind with different kinds of antigen. And polymorphism also we are able to see, or variety of the genes we are able to see here in this cleft only where the antigen binds. So as these two, alpha 1 and alpha 2, are responsible for making that peptide, that cleft for the peptide binding, so you can see the variability is also highly present in alpha 1 and alpha 2 only, in these two subunits only, not in the alpha 3. Okay, alpha 3, it has to bind with the CD8 CD8 molecule which is present on the T cells. Okay, so that binding should not be, it cannot differ, otherwise it will not even recognize its own T cells. Okay, so making that specific binding with alpha 3, that is not being changed, but for making or binding with different kinds of antigen, here there is a lot of polymorphism and variety of nucleotides are present. So that is what is being shown here, and that is how, so these are the places where there is a lot of polymorphism is found. Okay, similarly here also for beta chain it has been shown, beta 1 and beta 2. So here also for beta 1 only there is a lot of polymorphism or variability in these regions are reported. Okay, so the protein products of this MHC class 1 and class 2 genes, so they are highly divergent. Okay, so let's understand. So this is from the very beginning ancestral MHC gene is being shown. So in the beginning ancestral MHC gene, it undergoes a gene duplication and divergence. Okay, so there is only one gene. Okay, so then further it underwent the gene duplication, as I was saying, gene polygyny. Okay, the polygyny was there, so then there is a duplication event. Okay, and divergence event. Now you can see there are multiple MHC genes are there on both of the alleles. Okay, after that there is a gene conversion between the misaligned chromosomes during meiosis. Okay, so what happens during meiosis, there is a crossover happens. Okay, which is between the homologous genes. So even though, so they all are same genes, right, but there is a difference, they are divergent because of the presence of few nucleotides which are, due to which they differ from each other. Okay, but a majority of the portion remains the same, and that is why when there is a crossover takes place, uh, this particular one can align with this one, because that is also kind of homologous to it. It's not exactly identical but homologous. Okay, so if there is a misaligned chromosomes during meiosis, there is a gene conversion event, and after this segregation you will have something like this. Okay, this gene is now changed. What is it was initially, it is changed due to the crossover due to misalignment. Okay, so now the multiple MHC genes I'm having, then the gene conversion between the misaligned chromosome during meiosis, it generates variety there itself, and then separated chromosomes after meiosis, and due this multiple conversion events during evolution, we are having a variety, and these many in numbers of the MHC genes. Okay, that has been inherited to the progenies as well, and all are being codominantly expressed. Okay, and also along with the gene conversion, genetic recombination is also there, which can create new MHC alleles. Okay, so here you can see there is a strain 1 and strain 2, which is polymorphic regions in allele from the closely related strains. Okay, you can see everything is same except the last portion. Okay, and the position of polymorphic regions in one domain of the MHC. So here also you can see where, so the white region is quietly conserved, and this colored regions are the one which are having polymorphism. So that also you can see at which region it is having the polymorphism. Okay, here also in the peptide binding cleft majorly, there you can see, and here polymorphic regions in allele from distantly related strains. Okay, so here you can see there is a lot of difference between two strains. Okay, they are not same. Okay, and there is a lot of polymorphism, there are a lot of differences between each other, between two distantly related strains, and here these are the closely related strains. Here you can see with the gene structure, you will see if you will see, the white regions are the conserved regions and these two regions are the variable regions, but these are closely related strains, they are kind of identical to each other. However, here the two strains of and showing the alleles which are distantly related strains, and there is a lot of polymorphism being observed. Okay, so here you can see the MHC polymorphism. It affects the antigen recognition by T cell by influencing both peptide binding and the contacts between T cell receptor and MHC molecule as well. Okay, so we are able to see these polymorphisms are being present in these alpha folds also. Okay, so here in the beta sheet your peptide binds. Okay, where this cleft is there in the beta sheet region, there the peptide binds, and these alpha folds they interact with the T cells, T cell receptor. Okay, and polymorphism we are seeing in both of these uh, foldings or in the structures. Okay, so what we can say here, so as there is a different gene or different allele, or there is a polymorphism in those sequences, so the ultimate structure of the MHC will also differ. Okay, that will create structural variability. Okay, so not only the binding of antigen, also the binding of this MHC with T cell receptor will also be affected. So here you can see, um, due to variability, this MHC variant is able to bind with this antigen perfectly, antigen X, but this particular MHC cannot bind with the T cell receptor here being presented here on the T cell. Okay, because of the structural variability coming due to the polymorphism in this particular variety of MHC. Okay, so here it can bind to the antigen but not with the T cell receptors. Okay, here in this case, also you can see there is a compatibility with this MHC with the T cell receptor, but not between the antigen that is it is able to bind with the T cell receptor. Okay, so this variability decides both the antigen binding, also its binding with the T cell receptor, which is best in this case scenario. Here this MHC molecule, this variant of MHC, it can perfectly bind with the antigen and also with the T cell receptor. So both way it is binding, and that's antigen presentation is happening and there is a recognition happening. Okay, and that's how the downward further uh, processing will start in the T cell. Okay, so also there is a distinct subset of T cell that bears an alternative receptor made of gamma and delta chain, that also being shown here. This is TCR, which is having gamma and delta. Okay, and that is able to bind some, not that is not uh, processing the antigen or recognizing the antigen that is being presented them by MHC. However, they bind to the antigen with some different machinery, but these are very not that many in the number, they are very less in population, but they cannot bind to the antigen being presented by MHC. Okay, along with that we also have a pool of alloreactive T cells which can recognize non-self MHC molecules. Okay, so these alloreactive T cell recognizing non-self MHC molecules are also very abundant. That means um, in my body whatever MHC molecules are being generated by my immune cells, okay, uh, our T cell can only recognize those antigens only which are being presented to our MHC system. If not, then they will reject that. Okay, so this plays an important role in the graft rejection process also, this alloreactive T cell, where we are having um, different graft which is implanted in our body during transplantation and everything, where different sets of immune cells are there and they are having a different MHC. Okay, and with the help of that they present the antigens. Okay, so the there are a set of T cells which are known as alloreactive T cell. They also recognize non-self MHC molecules, and these are being present. Okay, and along with that there are a T cell population which respond to super antigens. So super antigens means that antigen which does not require to be presented by the what you can say the MHC molecules. Okay, for example, this one you can see, this antigen is not being presented as this one. Okay, it is only bringing both T cell receptor and MHC molecule together, but not being presented in a conventional manner. Similarly, this one also not being presented in the conventional manner, but that also these are called as super antigen, and they can initiate the T cell response. Okay, so these super antigens are being utilized in the laboratory processes where you want to enhance the T cell response process, and there you put the super antigen along with your own antigen that will enhance the T cell response and activate the T cells. Okay, so these are staphylococcal enterotoxin, SEs, and TCST1. These are called as super antigens, and in laboratory these super antigens are being used along with the original antigen to enhance the immune response or activate the T cells of the immune system. Okay, so to summarize, the protein products of MHC class 1 and class 2 genes, they are highly polymorphic. These MHC polymorphism affects the antigen recognition by T cells by influencing both peptide binding and the contact between TCR and MHC. There are alloreactive T cells which can recognize non-self MHC molecules, are very abundant, and there are many T cells that respond to super antigens as well. These MHC polymorphism extends the range of antigens to which the immune system can respond. Okay, and many proteins involved in the antigen processing and presentation are encoded by genes within the MHC. So let's see that. So the major histocompatibility complex. So let's see. So antigen presentation function both in arming effector T cells and triggering their effector function to attack pathogen infected T cells. So here you can see there is a cell. It is having this secretory vesicles. It is having these endosome, lysosome machinery, autophagosome machinery, and that is how it engulf the exogenous antigens and process them, and then it fuses also with this lysosomal machinery also. Okay, so let's see. So cytosolic, there are cytosolic pathogens, there are intravesicular pathogens, and there are extracellular pathogens. So these two, intravesicular and extracellular pathogens, they are being presented by MHC class 2. So they are being processed or degraded in the endocytic vesicles which at low pH, which is at the acidic pH. Okay, and they are being presented to the effector CD4 T cell, and here these one, they can activate to kill intravesicular bacteria and parasites, and activation of B cell to secrete the immunoglobulin to eliminate extracellular bacteria or toxins. On the other hand, apart from the extracellular antigens, we are having the cytosolic pathogens, for example from viral or tumor cells. So they are having the pathogen which is cytosolic, and they are, these peptides, they bind to MHC class 1, and they are being presented to the effector CD8 positive T cells, and the effect of this will be the death of the antigen presenting cells. Okay, so here MHC class 1 and MHC class 2 molecules. So MHC class 1, MHC class 2, here you can see this presents the MHC class 2, it presents the extracellular antigen. Okay, the extracellular antigen being engulfed, and then they are, so before binding to the or loading the antigen, these MHC class 2, they are bound to a molecule called CLIP. This antigen, they and also the foreign antigen that also come in a vesicle, they both combine. When the vesicle merge, CLIP is unloaded and the degraded antigen will be loaded on the MHC, which is being presented on the surface with the MHC class 2. Here is an example of cross presentation of exogenous antigen by MHC class 1. So we know exogenous antigens are being presented by MHC class 2 molecules, but there is also one cross presentation which is done by the dendritic cells. So here this particular cells which has engulfed this antigen, okay, that is being engulfed here in the phagolysosomes, and then these phagolysosomes, inside that they load these antigens on the MHC class 1 and express them and present them on the surface with the help of MHC class 1. This is a cross presentation example, okay, where the dendritic cells takes part. So this is also an extracellular antigen, but it is being presented with the help of MHC class 1 molecule, which is called as cross presentation by dendritic cells. Okay. So the T cell, they orchestrate the cell mediated immunity and regulate B cell response to most antigen. So here you can see this is a viral infected cell. Okay. So the virus is releasing its genetic content into the cell, and here with the help of that, inside the cytosol of the cell, the transcription and the translation will start. Okay, the genetic material of the virus start the translation, and as the translation starts, there is a peptide fragment of viral proteins bound by MHC class 1 in the ER. Okay, so in the endoplasmic reticulum only there are these MSCs are present, where it encounters these antigens, and then they load themselves with the antigen, and then in the vesicle form goes on to the surface and present this particular antigen. Okay, so the peptides are generated from the ubiquitinated proteins in the cytosol by the proteasome. So not the full protein is being presented on the MHC surface. So what happens? So as we have seen at the MHC structure also, okay, here you can see there is a cleft. Okay, so here in the previous class we have seen that in this cleft only a certain length of peptide can accommodate. Okay, and that there also the residues there are certain residues which remains conserved. Okay, so the entire protein cannot be presented, but a peptide fragment can be presented. So to do that fragmentation, there are some machineries. So there are some machineries to do that conversion. So here you can see, so this is a proteasome. This is called a proteasome, which is having these 20S 120S core combines with 219S regulatory caps to form a proteasome in the cytosol. So the protein which is being synthesized in the cytosol, so that is being degraded with the help of this proteasome. Okay, this is the proteasome structure. It is a barrel kind of structure, and the protein which is synthesized in the cytosol gets ubiquitinated. So polyubiquitinated proteins are bound by the 19S cap here and degraded within the catalytic core here in between this, releasing peptides into the cytosol. So there these peptide fragments are being generated. Okay, here this is the transverse section of that particular proteasome. So here inside this barrel structure you can see there is a catalytic chamber in which the catalysis will take place and degrade the protein into the peptide fragments. Okay, so this is a newly synthesized MHC class 1 molecule are retained in the endoplasmic reticulum until they bind a peptide. Okay, So here you can say the partially folded MHC class 1 alpha chain binds to calnexin until beta 2 microglobulin binds. So we know MHC class 1 are having alpha subunit and beta subunit. Okay, so alpha, so you can see this is alpha subunit which is present there in the endoplasmic reticulum. So alpha 1, alpha 2, alpha 3, this is there, and here it this alpha chain binds to calnexin until the beta 2 microglobulin binds. So this is bound to the calnexin in the ER. Okay, then there is the MHC class 1 uh, alpha beta 2 this microglobulin complex is released from the calnexin. Here it released from the calnexin and then binds a complex of chaperone proteins, so calreticulin and ERP57. Okay, so these are the chaperone proteins, calreticulin and ERP57. It binds with these two. Okay, so as soon as beta comes and binds, it releases from the calnexin and binds to the calreticulin and ERP57, which are the chaperones. Okay, so then what happens, there you can see this is also there, uh, this tap protein. So what happens next is the cytosolic proteins and defective ribosomal products, okay, DRIPs, okay, they are degraded to peptide fragments by the proteasome. Okay, so here you can see the proteasome machinery, it is degrading. So there are generated, these strips are being generated, and TAP delivers the peptides to the ER. So this TAPs, it helps in delivering the peptides to the ER. So from cytosol it is going to the ER with the help of this transporter called TAP. Okay, so a peptide binds to the MHC class 1 molecules and completes its folding. Okay, and the MHC class 1 molecule is released from the TAP complex and exported to the cell membrane. Okay, so there are these proteases are also present, LMP1, LMP7 and MXC L1, that does that proteasomal degradation to make those conserved residue also, which can bind to the cleft of the this MHC class 1. Okay, so this is what happening. So here the antigen comes and bind after the the proteasomal degradation of the ubiquitinated protein, which is being transported with the help of TAP and being loaded on this particular MHC molecule. Okay, and once the peptide binds the MHC class 1 molecule, and this folding gets completed, and this class 1 molecule released from the TAP and then exported to the cell membrane. So for the MHC class 2 complexes, the peptides are generated in the acidified endocytic vesicles from proteins obtained through the endocytosis, phagocytosis and autophagy, all of three methods, the endocytosis, phagocytosis and autophagy. Okay, so for MHC class 1, as it presents the endogenous antigen which are being synthesized inside, which is usually a viral antigen. Okay, this is the process of presenting that on the surface with the help of MHC1. And for MHC2, they take up the peptide which are being, like the exogenous pathogen, take them up with the process of endocytosis, phagocytosis or autophagy, degrade them and then present them on the surface with the help of MHC2. Okay, so this is the extracellular antigen, exogenous antigen. So antigen is taken up from the extracellular space into the intracellular vesicle. So in early endosome of neutral pH, the endosomal proteases are inactive. Okay, then the acidification of vesicle activates the proteases to degrade the antigen into peptide fragments. Here you can see there is a degradation of this peptide protein into the peptides has happened. Okay, and then the vesicle containing the peptides fuse with the vesicle which contain the MHC class 2 molecule. So there is one vesicle which is containing the peptides, the degraded peptides of the foreign antigen, and there is one vesicle which is carrying the MHC molecule. They both fuse with each other and then the loading of this peptide on the MHC class 2 happens. Okay, so before binding to this particular antigen, these MSC class 2 molecule is preoccupied. So it occupied with a clip molecule. So what happens initially? So the invariant chain directs the newly synthesized MSC class 2 molecule to acidified intracellular vesicle. Okay, so here you can see initially there are this trimerization of this particular MSC class 2 is happening with the help of this particular protein. Okay, this is a transmembrane protein. This is a structure which is called clip. So then what happens? This is the cytosol, this is the ER. So the invariant chain, okay, invariant, this IA, it's invariant chain. It binds in the groove of MHC class 2 molecule. Here you can see it binds in the groove of MHC class 2 molecule. So then this invariant chain is cleaved initially to leave a fragment bound to the class 2 molecule and to the membrane. Okay, here, and the further cleavage leaves a short peptide fragment clip bound to the class 2 molecule. So here it is preoccupied with the clip. So then what happens? So the invariant chain forms a complex with MHC class 2 molecule blocking the binding of peptides and misfolded proteins. Here at this step, this is what is happening. So then the invariant chain is cleaved in acidified endosome, leaving a short peptide fragment clip. It is still bound to the MSC class 2 molecules here. Okay, this is what is happening. Then there is a endocytosed antigens. This is a foreign antigen which is coming by endocytosis. They are degraded in the peptides in the endosome, but the clip bound peptide blocks the binding of the peptide to the MSC class 2 molecule. Here you can see this blocks the binding. Then what happens? So this HLA-DM, okay, so here in this vesicle along with the MHC class 2 molecule there is one more gene which is or protein which is named HLA-DM. So it binds to those class 2 MHC molecule and then releasing clip and allowing other peptides to bind. So MSC class 2 molecule then travels to the cell surface. So this particular binding of HLA-DM to MSC class 2 molecule releases the clip and allows other peptides to load on MSC class 2 molecule. Okay, so there are studies which shown that if there is a mutation in this HLA-DM, there is a difficulty or the antigen processing of the MHC class 2 is hampered. So that is why this interaction is very important for the proper antigen processing and presentation by MSC class 2 molecule. Okay. The cessation of antigen processing occurs in dendritic cells after their activation through reduced expression of MARCH1E3 ligase. Okay. So in immature dendritic cells, okay, the MARCH1 it ubiquitinates the MSC molecule targeting them for degradation. So usually what happens if in one particular scenario you put one macrophage, okay, and then on the surface of that you will find MSC molecules but not many, but then when you infect the culture with one pathogen, then you'll see the expression of that MHC molecule on the surface of macrophages increases. So how that regulation comes? Like in the presence of the pathogen and in the absence of pathogen. So in the absence of pathogen there are less MHC molecules being expressed on the surface of the antigen presenting cell, and the presence of pathogen there are the expression of MHC on the surface increases. So how that happens? So what happens here? Here you can see in this vesicle there are 3 MSC molecules, but there is this particular the MARCH1 protein which is the ligase. So it ubiquitinates the MHC with which it is associated. So the ubiquitination is the sign for the degradation. Okay, so when ubiquitination takes place, it degrades the MHC molecule. So out of the two, only one could go and express on the surface. Okay, but when it encounters a pathogen, so that pathogen with the encountering the pathogen, the activation stops. Okay, So the activation of this TLR toll-like receptors will take place, the dimerization and activation. Their activation gives a signal to the cell, the via TLR signaling, which inhibits the transcription of this MARCH1 gene. Okay, so the transcription of this MARCH1 gene gets down regulated. So MARCH1 was the one which was degrading the MHCs. So if the MARCH1 transcription is down regulated, so once, and you can see the half life of this MARCH1 is 30 minutes only. Okay, so here after giving the pathogen, after 30 minutes, whatever MARCH1 was there, that will perform its function, but after 30 minutes you will see MARCH1 is no longer present, because there is a transcriptional deregulation of this MARCH1 is happening, and due to that it cannot inhibit the expression of MSCs on the surface. So now due to the inactivation of MARCH1 gene, due to the activation of TLR signaling in the presence of pathogen, allowing the expression of more than one MSC on the cell surface. So that is how the MSC expression on the cell surface increases in the presence of an antigen presenting cell with the presence of any pathogen. So this is it. Let's take the questions related to the today's whatever we have gone through. So here you can see the first question is, the class 1 MSC gene that encode for membrane bound glycoproteins are found in. So we have read in the previous class only, class 1 MSC genes, they present on all nucleated cell. So except RBC, so RBC is non-nucleated, except that all nucleated cell express the class 1 MSC gene. However, class 2 MSC gene, they are being present only on the professional antigen presenting cell, which are B cell, macrophages, dendritic cells. For this question, the class 1 gene is encoded for membrane bound glycoproteins are found in all nucleated cells. Okay, so the correct option will be the first one, which is this one, the all nucleated cells. Okay, so polymorphicity of this class 1 and class 2 of MSC genes occurs due to, okay, so there are these four options given, which one could be true: gene conversion, gene recombination, or PTMs. What is the reason of arising polymorphicity in MSC class genes?
Option D, both A and B.
Yes, for polymorphism, they undergo gene conversion as well as the genetic recombination. Yeah. So the T cell which can recognize non-self MHC molecules, they are called as what? They are abundant in our system. They are called as
Alloreactive T cells.
Yeah, they are alloreactive T cells. Yeah, this E3 ligase called March1. Okay, this is, so what is the, we have to choose the true statement. So first statement is, this E3 ligase March1 is expressed in B cells, dendritic cells, and macrophages. Is it true? So all these three, what are these? These are APCs, right?
APC.
Yeah. And these antigen presenting cells, they express which MHC on their surface?
MSC1.
MHC1 is being expressed by all nucleated cell, but these APCs, they express MHC2 on their surface. Okay, MHC2. MHC2 is found only on the surface of these cells: B cell, dendritic cells, and macrophages. Okay, so this is APC which are expressing MHC type 2. Okay, and they present the antigen with the help of MHC type 2. Okay, so the machinery that we have read for this one, for the E3 ligase March1, that is for the expression of MHC class 2. Here you can see an MHC class 2 is present on the surface of B cell, dendritic cell and macrophages. So E3 ligase March1, it helps for the expression or the surface expression of MHC class 2. MHC class 2 expresses on these APCs. So first option is correct. This E3 ligase March1, it resides in the membrane of recycling endosomal compartment. Okay, this is also correct. Okay, it resides in the endosomal compartment, which is shown here. This is recycling endosomes where MHCs are being present, and there this March1 resides. Okay, and third one is ubiquitinates the cytoplasmic tail of MHC class 2 molecule. That is correct. So with the help, it ubiquitinates, here you can see, it ubiquitinates the cytoplasmic tail of MHC and thus degrades, leads to the degradation of these MHCs. These E3 ligase March1 gets inactivated only when there is a activation of this TLR signaling, when there is a pathogen is present, and then there is a silencing of this gene March1, and thus it can no longer ubiquitinate and thus degrade the MHC class 2 molecule, and thus the expression of MHC class 2 molecule on the surface of the antigen presenting cells increases. So the third option is also correct. So all the three options are true in this aspect. Okay, so MHC class 2 present antigen that are obtained by, so we know MHC class 2, it present the extracellular antigens. So they are obtained by which process? Which?
All of them.
Yes, it presents, engulf the antigen with all these three processes. So how the T cell receptor is different from B cell receptor? Okay, so the T cell TCR does not contain a variable region like BCR. Is it true?
TCR is having a variable region.
Yes. Yeah. So here, if you remember the image, so BCR is nothing but the antibody. Okay. So BCR, it is having the variable region, B cell receptor having variable region. Similarly, the T cell receptor is also having the variable region. Okay, so this is not true. The TCR is never secreted in plasma. So unlike the BCR, which is the B cell receptor, which is nothing but the antibody, so there are two forms, one which can be secreted also. So the B cell receptor can be secreted uh, uh, in the plasma, but TCR cannot be secreted in the plasma. It remains anchored with the T cell. So this is correct. It is never secreted in the plasma. So the two chains of the TCR are not linked. No, they are linked. They are linked with each other, these two chains. So the correct option is TCR is never secreted in the plasma. Okay. So stimulation of the naive cytotoxic CD8 positive T cells into activated CD8 positive T cells is a consequence of cross presentation of exogenous antigen. Okay, cross presentation. So which among of the following is most efficient cross presenting cell? Cross presentation we saw in case of dendritic cell, they are the most efficient cross presenting cell. So it is a exogenous antigen taken by the dendritic cell but being presented with the help of MHC1 molecule to the CD8 positive T cells. So the correct option is dendritic cell. Okay, yeah, so this is, if there are these many numbers of DRA and these many numbers of DRB genes present in a human, how many types of HLA class 2 could be expected? Okay, so HLA class 2 is nothing but the MHC class 2. So they are formed with the combination of alpha and beta. So this is alpha subunit, this is beta subunit. MHC subunits, alpha and beta. And if there are 345 genes for alpha, 45 for the beta, and how many total could be expressed? So as there is a combination of both alpha and beta is being expressed there, and all are being expressed, so the multiplication of these two numbers will be the correct option. So both the combination, both the number multiply together, and that's that many combination of class 2 HLA can be expected. Okay, so multiplication of these two number will be the correct option. Okay, so molecular chaperone involved in the assembly of class 1 MSC. Okay, so there are certain chaperones, right, which are involved in the assembly of class 1. What are those molecules? What are those?
Both A and C.
Yeah, this is calnexin, which binds to the MSC class 1 when there is only alpha subunit is there. When the beta subunit binds, calnexin releases, and then the calreticulin and the another chaperones come in the picture, and they bind and allow the folding. Okay, so calreticulin helps to stabilize the complex, this one, for peptide loading by binding to the glycans on the MSC class 1 molecule. So, and the ability of calnexin to protect the and retain the class 1 assembly intermediates is likely to contribute the efficient intracellular formation of class 1 complex peptides. So the correct option will be these two. Okay, find the correct sequence for antigen presentation for exogenous antigen. So which one would be correct? Okay, first one is, it says first one is says binding of invariant peptide to MSC. Okay, so this one, binding of invariant peptide to MSC. So in the beginning an invariant chain is bound to the MSC. Okay, true. Then it says cleavage of the invariant peptide. So after the binding, the invariant peptides gets cleaved. Okay, and then there is only one fragment which is called as clip, which remains associated with the MSC. Okay, so after that there is a removal of that cleaved peptide. Okay, so that so when there is a invariant peptide is there here, so here that invariant peptide remains, so that invariant peptide doesn't allow the binding of the foreign antigen. So first is this invariant peptide, the long chain binds, then there is a cleavage of this invariant peptide, and then also when there is a antigen comes, this invariant peptides it removed with the help of another protein which is called as HLA-DM. You can say HLA, with the help of HLA-DM, this peptide it gets removed from the MHC, and the processed antigen is being loaded on the MHC. Okay, and then it is being processed, or it is being loaded onto the MHC. So this is correct. Binding of the invariant peptide to MHC, then cleavage of the invariant peptide, then removal of that cleaved peptide, and then the binding of the processed antigen. So the first option is the correct one. Is it clear? It's clear, or is there any confusion? Do you have anything to ask from this session? Okay, if there is no question, I'll conclude the session for today. Okay, I'm concluding the session. See you again next Tuesday.